Thank you, Marge. Tēnā koutou katoa, ko Araki te maunga, ko Waimakariri te awa, ko Hayley toko ingoa, nō reira tēnā koutou, tēnā tātou katoa. So I'm just welcoming you all to this talk today, and I just describe myself. So my mountain is Aoraki, my river is the Waimakariri, my name is Hayley, and I am based in Lincoln, Christchurch. So today I'm going to talk about the foliar microbiome of myrtaceous species and some implications for myrtle rust infection. I'm the spokesperson, but by no means did substantial parts of the work. So my co-authors are down there on the bottom of the screen, and I'll acknowledge them now, but also as I go through this talk. So this talk is about work that we've done as part of the Beyond Myrtle Rust project. So you're probably all quite familiar with what the microbiome is, but just like humans, plants host a large community of microorganisms on them and in them. So when we look at plants, we sort of see the macroorganism, probably with insects or whatever have you on them. But what is the reality is actually that there are a large number of microbes living on and in the plants. And that living community, the plant and the microbe, is termed a holobiont, and it is that ecological unit that interacts with the environment. The microbes that live on or in the plant are collectively termed the microbiome, and it's multiple taxa, both bacteria and fungi, and often protists and other organisms. So when Myrtle Rust invaded in around 2017 into New Zealand, so this is not new to anyone, we know that it affects foliage, flowers and fruits, and in New Zealand we have approximately 30 native myrtaceous species. There was field observations that a single twig has foliage of different susceptibility. So the young, new growth, both on the flowers, the fruits and the leaves, is quite susceptible to myrtle rust, can be infected and die. And it raised the question of whether there was any differences, firstly, between the microbiome of the young new season's growth and last season's growth, and whether that phylloplane microorganisms could influence the susceptibility to myrtle rust. And if it could this be a source of novel biocontrol agents? So in our Research AIM project, we had a title called Novel Mitigation Technologies, and the focus was to understand the role of microbial community related fitness, phenotypes and tolerance to Austropuccinia psidii. And this involved a workflow which had multiple strands. The first one was to generate some collections, so a culturome. These were the organisms that we would ultimately like to test. Accompanying that, we characterised the complete foliar microbiome. And that's because although some of the organisms will be culturable, not all of them will be. We then wanted to compare the microbiome community structure across hosts, across sites, and particularly between new season's growth and last season's growth. And then from the culturome, we wanted to identify any organisms that had potentially antagonistic potential towards myrtle rust. And this involved quite a lot of bioassay development so that we could rapidly get through quite a large number of isolates. And the ultimate goal was to see if we could augment plants by replacing or adding new microorganisms that would colonize the phylloplane of the young tissue and help resist infection by myrtle rust. And what I'm going to talk to you about is our progress on the way there. So Kirsty Boyd-Wilson kind of led the culturome generation. So we looked at culturome generation from 10 plant species, and you can see them there in the table with the blue title. However, we focused most of our effort on 3 iconic species, which are Metabarcoding excelsa, which is our pōhutakawa, Leptospermum scoparium, which is mānuka, and Lophomyrtus ballata, which is ramarama. These had very different leaf morphotypes, and you can see the leaf morphotypes there. And we ended up culturing a large number of organisms. We have a collection of more than 2,000 epiphytes and endophytes recovered from these plants. They were put into pure culture and subcultured and identified using basic ITS sequencing. From that information, often for the bacteria it was only to the genus, but for many of the fungi it was to the species level. We placed them into 5 groups based on correlation with known literature, so whether they were likely or whether they were taxa with similar identities that were known hyperparasites, known biocontrol agents, known growth promoters. We put actinobacteria in their own class, and then others, which captured everything we didn't classify. Accompanying that, and this is where the samples were taken in order to produce that culturome, we looked at the complete foliar microbiome from 5 locations around New Zealand on the 3 hosts specifically. We took 3 branches from each of 5 plants per species, and we separated these into stem and leaf samples, and we compared the phylloplane using Illumina metabarcoding. And so you can see on the right-hand side here the locations around New Zealand that we went to. So 2 sites in the South Island, quite close proximity together, and 3 sites in the North Island. And you can clearly see the difference between the young seasons or new seasons growth of pōhutakawa, which is the light green leaves on the far right, and the darker leaves of the last season's growth. So it was fairly easy to distinguish between new seasons and last season's growth. We undertook microbial community analysis using Illumina metabarcoding. And the first question we wanted to know was, we'd taken 3 branches from each of these plants. Were they substantially different from each other? And if they weren't substantially different from each other, could we then pull the samples? And this work was undertaken by Pretty Panda. And what she showed was that the branches on a single tree were not significantly different in terms of their diversity between each other. So we pulled our samples and did metabarcoding on the pulled samples. And this is one of the pieces of information that we found. So the phylloplane composition is distinctive between the plant species for all 3 taxa. There was some overlap, so you can see here the NDS plot for mānuka, pōhutakawa, and ramarama, and you can see a large diversity for mānuka, which encapsulates the pōhutakawa diversity, and then the ramarama, which is distinctly different. Accompanying that, we did other analyses, such as an LDA score, which looked at, on the left-hand side, a bunch of, in this case, different fungi, and then whether they were common or less abundant on any one of the 3 hosts. So you can see that Hortia species were very abundant on pōhutakawa, but not very abundant on ramarama. So that's how you interpret the red, blue and beige colours. And this is just the LDA score as their relative abundance. We then also compared the foliar microbiome across young and old tissues. So we had, I don't know if you can see this, quite small, but young stem and young leaf as the top 2 bars, then old stem and old leaf as the bottom 2 bars for each of the panels, which are mānuka, pōhutakawa and ramarama. And what you can see is that there is some difference between young and old tissues, mainly in relative abundance of organisms, not particularly as strong in presence or absence, although in the pōhutakawa we did see Capnobotryella as quite dominant in the older tissues, and that correlates quite well with the sooty mold that we saw often on those plants. Then we also started to look at the antagonistic potential. So we had this large culture collection. We had seen that there were differences between old and new foliage, and we wanted to know whether the microorganisms from the older foliage, the last season's foliage, would be able to offer some protection against Myrtle Rust infection. So we chose to do this initially on pōhutakawa, mainly because it has lovely big leaves on which to apply said microbial organisms, but also nice scoring, and we're able to recover organisms from the phylloplane quite easy. So we had new leaves which were inoculated either with the carrier, with a mixture of 3 bacteria that were prevalent in the older tissues, a mixture of 5 bacteria, that were prevalent in the older tissues, a bacterial and fungal mix, a mature leaf wash, which is a wash we took from mature leaves, so that's kind of the background control, and then an untouched control. We spray inoculated those organisms onto the plant and left them for 1 week to stabilize, and then we challenged them with the rust. We scored the leaves at the end of the experiment, and we also sampled leaves over time and did a post-challenge metabarcoding to see what was actually happening to the phylloplane communities after we had put new organisms onto that phylloplane. We didn't get any difference in rust severity, although some plants were more or had less rust than others, and it seemed to be a bit intermittent. We didn't get a massive amount of rust, so it may have been that the challenge was also not as strong as we potentially would have liked. But what we wanted to see was using deep sequencing tools, what was actually happening on the surface of that plant. And this is what we saw, and this work was done by Justine Louis, who is a new scientist in our group. And what Justine looked at was the populations, in this case I'm showing the bacterial populations on the leaves over time. So at the bottom you can see the days, so day 1, day 3, day 7, day 14, day 21, and you can see the untouched control, the carrier control, the mature leaf wash, and then the bacterial application. And what you can see is that in the untouched control, nothing changes very much over time. With the carrier only, there is some change over time, and that's possibly because there's some type of nutrient source being put on the surface of the leaves. And the mature leaf wash, you can see by day 7 that Pseudomonas is starting to dominate the community, and that tapers off at day 21. And what you can see with bacterial wash is when we applied Pseudomonas, which was one of the 3 bacteria that we did apply together with Curtibacterium and Sphingomonas, you can see that from day 1, the Pseudomonas dominated the organisms on the phylloplane, and this was quite different to the untouched control. So although we had not seen a change in rust severity, what we had seen was that when we apply a bacterium, particularly Pseudomonas, to the leaf surface, it was able to establish and persist, and it became the dominant taxa over that 21 days. And that was quite critical information to support the delivery of a bacterial biological control agent to the leaf surface. It suggested that out of the 3 that we tested, and we only tested 3 genera, that Pseudomonas was quite suited to the leaf surface. So we carried on with this. So we had our 2,000 odd isolates placed into the 5 groups, and we decided to undertake some batch testing. And we do this batch testing in a PC2 environment. So you can see, I think that's Kirsty, not sure, it could be Suni there, standing there in front of our little humidity tents, looking at the plants and scoring the rust severity. So what we did was we challenged we applied the candidate strains to Lophomertus ballata now, so this is ramarama, then challenged them with the rust 7 days later. And these bacterial isolates that we used had previously gone through a high throughput screen in a germination assay, so we'd already attempted to pre-screen from the 2,000 to get around 150 that we wanted to test through batch testing. And this is an example of the results we saw with the batch testing. So we're looking for novel control agents in the culture collection. We're working on Lophomertus ballata as a highly susceptible host. We are using bacterial isolates from a germination assay. And on the far right, you can see an example of data from a prior germination assay. So on the left-hand side, you can see a bunch of numbers. These are unique numbers given to each of the bacterial strains. And down the bottom, you can see the rust-only control and the percentage of spores germinated. So this is a germination assay of Austropuccinia psidii. When we add the bacteria, you get various levels of inhibition of rust germination, with isolates 776 to about 881 starting to inhibit rust germination. So we used these in batches at 10 to the 5 cells per mil added to the plants. So for example, BA, which is shown there in the dotted square, is our backstar control, and that is the best commercial biological that we have found so far. So we use it as a positive control in our assays. Then PS, which is a mix of 4 Pseudomonas isolates, was giving equivalent control to backstar. I will point out that this is delivered at a lower overall cell concentration than backstar, and it's delivering as good protection. It's also a mixture of 4 isolates, and we don't know whether all or just one of those is delivering the effect. And similar for the SS mix, which also delivered reasonable level of control. So we went through batch testing for a large range of isolates, and we did 5 experiments on these. So each of these experiments represents batch testing of different microbial collections. We had varying success with our ability to cause infection, and we suspect that this is to do with temperature fluctuations in our glasshouse over the seasons. So you can see in August we didn't get a very good range of infection, with very few of them being significantly different from each other. Whereas in February we got a very good range of infection with backstar showing quite a lot of control, and the PSSP4 group also showing quite good control. So what we did was we took our best from that and we decided to try them individually. Unfortunately in April we didn't get very good infection, and this did not work very well. So this is a back to the drawing board, trying again once we've got better control over, we believe, temperature in our glasshouse. However, we did complement that with a twiglet assay, and this was developed by Mānuka Joshi and Suni Ching. And these are little twiglets of ramarama on water agar plates. And what we're doing is we're using the bacteria that we're interested in as single strains in concentrations similar to what you would see in the backstar commercial preparation, and we are spraying them onto the surface of these twiglets and then challenging them with Austropuccinia psidii. And what you can see here in the graph is the probability of any disease score for each of the treatments. So the disease score across the top is 0 to 4, 4 being a high level of infection and 0 being no infection. And what you can see is that Bacillus species 776 had a high probability, probably about 75%, of giving a 0 score. So it was an isolate that helped protect against infection by Austropuccinia psidii. Similarly with Pseudomonas, and you can see the backstar down there, was either giving a 0, no infection, or a score of 1, but the Bacillus was outperforming it in this case. Again, it should be noted that our control, which is tween, so this is just the carrier delivered with Austropuccinia psidii 7 days later, and what you can see is that our infection score was around a 2. So again, it wasn't huge amounts of infection. We'd like to improve our ability to cause infection on these plants. So again, we'd like to repeat that with a higher concentration of the novel agents. Accompanying this, we also have a PhD program that we started, and this is because we had a whole bunch of potential hyperparasites or mycoparasites that needed more work. It was more than just batch testing. We wanted to look at their ability to infect Austropuccinia psidii and their ability to kill it, essentially. And this work is being done by Dante Gu, who is undertaking that PhD program with Arian Jones and Suni Ching. And she has 4 objectives. So looking at the microbiome associated with Myrtle Rust infection, screening and identification of the organisms she recovers, and then evaluating their biocontrol efficacy and looking at their mechanism of action. And if she has time, we'd like to know where these mycoparasites are coming from. They've probably not been co-imported with the rust. They're probably from our native Tolna species in New Zealand. So she undertook quite a lot of sampling, and I'd like to acknowledge Kirsty, Vlad, Michael and Stuart here from Scion, and particularly a big thank you to Scion, who have not only supplied us with some of their mycoparasites that they have identified, but have also been extremely helpful in helping us do the collections for Dan. So she's sampled in 4 areas or 4 locations and 8 sites. So she's created a collection of 192 isolates with 57 genera, and she's found both epiphytes and endophytes. And from these, she's identified some presumptive mycoparasites. So there was actually quite a large number of trichoderma species recovered, and Coleototricum, some cladosporiums, Alternaria, Clonostachys, Epicoccum, Paraphaeosphaeria, Phlebiopsis, and Sclerostagnospora. And she's probably going to sit with that number to move on to the next stages of her PhD, but we do have quite a few that warrant further exploration. So what's she going to do? She's currently undertaking the twiglet assay to confirm whether or not they are mycoparasites, and that will involve some microscopic examination of the infection of pustules, the taxonomic placement of the confirmed mycoparasites, and then selection of focal taxa to work on for her PhD. She also has paired samples of leaves infected by myrtle rust with an adjacent leaf uninfected by myrtle rust. And she's asking the question, does myrtle rust change the phylloplane microbiome? And where are these mycoparasites coming from? So can we sample from surrounding potentially non-mutaceous species to see if they also host these organisms? So what are the take-home messages here? So the take-home messages are that the phylloplane microbial communities are distinctive between plant species, that the young leaf phylloplane is receptive to the application of protective bacterial species, that spore inhibition assay and an auxiliary twig assay have been developed as high throughput tools, and then our batch testing has identified 3 strains that warrant more work, both with concentration, the timing with which we apply them, and looking at the extent of their phylloplane colonisation using deep sequencing technologies. We also have a putative mycoparasite collection produced by the PhD. And what you can see in the slide on the right-hand side is you can see the rust germinating quite happily without bacteria. And then on the left-hand side, you can see the bacteria inhibiting the rust germination. Ngā mihi. Thank you, and are there any questions?
Thanks, Hayley. That was really great. Audience, if you have any questions, just put them into the chat and then I will read them out. So I'm going to start with a question because I'm not sure if you mentioned this, but so the communities were different depending upon the species, but how about from the locations? Did you see differences from the locations as well?
Yes, there were some differences with the locations, Marge, but the host difference was much larger. So you did see regional changes, but it wasn't so much in the presence, absence of species, but it was more the relative abundance of colonisers. So everything was everywhere, but the relative abundance on different hosts varied.
Very interesting. All right. So there is a question. Please clarify whether all the bacterial genera are plant pathogenic.
So one of the things we do early on is check whether any of the bacteria are of known, I guess, pathogens. Knowing that for any one genus you can have both pathogens and non-pathogens. So the Pseudomonas that we have that we're interested in are not syringae. We have made sure of that. And most of them, I think all of the ones we're very interested in, we now have min-ion sequencing of. So we're pretty confident that they are not pathogenic. In that way, so we're basically looking at their full genomes and checking for pathogenicity islands, etc.
Sounds good. You also have some compliments coming in. Very nice talk, very interesting. I guess the other question I was just thinking about with the hyperparasite work, that sounds really fascinating too. Was there a particular strategy that that Dan had done to try and find these hyperparasites, or was she just sampling everywhere?
So what she's looking for is pustules that look parasitized. So these are derived from pustules. So she's collected pustules and then plated them onto agar and recovered the fungi that are growing out of the infected pustules. Now, we know that that may just be that something's accidentally landed on there, so we do need to confirm that they are actually mycoparasites before we move further. So we've done kind of a base Sanger sequencing to get an idea of what the taxa are, but we won't be doing a full taxonomic placement unless we've confirmed them as a putative mycoparasite. So that's how we've done it. We've isolated directly from odd-looking pustules.
Sounds good. Hi, Hayley. Have your commercialization team been alerted to the potential of the bacillus or pseudomonads? Sounds like somebody from Plants and Foods, somebody from your commercialization team.
So one of the things that we're very aware of is that these are microorganisms from Tonga species. So we do not own the rights to these species. So any development of commercial processes need to be done in partnership with Māori. So should we find something that has potential, then our very first response there is to reach back through to the location that we sourced it from and talk with the iwi there about what we have found, what the interest might be, and talk to them about potential next steps. But that's not something that we can strictly go ahead with just as we wish.
Yep, good. So is it possible for the bacteria to work curatively or only preventatively?
That's a really interesting question. And now that we've got some candidates, if we had another 5 years funding, that would be a fantastic question to look at. So we are really interested in timing. So we've been putting the bacteria on prior to infection or prior to challenge with Austropuccinia, but we have begun to ask the question, what would happen if we put them on once the plant was infected? And that's going to be part of some work that we're intending to do in the next year with some new funding that we've managed to obtain, is to look at whether we can limit or prevent infection once it has started.
Sounds good. So there's another question. A person notices that insect damage on pōhutakawa seems to bring more infection on young growth. Do you have any comments about that?
I haven't done any work on that. There could be a whole range of things that are happening, such as plant stress, that may make it more susceptible to infection. I'm not sure whether that has any influence on the phylloplane microorganisms, but it may well do. You know, insects bring their own gut microflora to feeding events as well. So that could potentially be an interesting thing to look at.
Yep, absolutely. And another question is that some endophytic bacteria are known human pathogens, especially in immunocompromised people. Is this something that you're going to consider in your work?
It's certainly not anything we'd want to be working with. I am aware that sometimes trichodermas do get that label. We need to be aware of that. We're certainly very careful when we're working with it. We would exclude any known human pathogens from the work, but many fungi are a problem for immunocompromised people. So it's important that our researchers working in this area are aware of that and excuse themselves from that work if they need to. But I think if it's with respect to the trichoderma, we will probably travel on with those at this point.
Yep. Yeah. No, it sounds good. And it's really fascinating. I mean, I think it's, you know, just, it's really been amazing just to see, like, you know, the outcomes and to see like how, like, even though you had 2,000 different organisms, and from that, 3 of them are promising. So that's, you know, that definitely reiterates the importance of casting the net
Large
wide to try and,
collections.
yeah, exactly, and to
Mm.
try and get to that answer. So if there aren't any more questions, I think that's it for today, audience. Thanks, Hayley, for your presentation. That's been
Thank you.
really awesome. So a video of today's webinar is going to be made available on the Beyond Myrtle Rust website in the next couple of days, and it's also going to be emailed to people who have registered. We're going to take a break in January, but then we'll be back in February with the webinar series. We're going to be hearing from Daryl Heron at Scion. So Daryl is presenting on the Jobs for Nature project. So keep a lookout for the webinar invite next year. And with that, happy holidays, everyone. Happy New Year,
Yes,
and
happy
thank you.
holidays.
Hi, Ira. See you next time.
Ka kite.